Hello, welcome to the HPP Innovation Week, and welcome to this session on the application of high pressure processing, HPP, to pharmaceutical and cosmetics products. I will be the first speaker of this session. I'm Carol Tonello. I'm Commercial and Application Director at Hyperbaric. I work for the company for almost 20 years, and I have a PhD in food microbiology, inactivation of microorganisms under pressure. I will give the first presentation, but I will be in this session, I will be also with three other people, and I'm very pleased to present now Iker Portugal. Iker is a shareholder of the company Licolab, and he will be with us to present this company and its use of high pressure of HPP high pressure processing for his very nice product. You are very welcome, Iker. The second
thank
speaker.
you very much.
Thank you. The second speaker is Pierre Combro. Pierre is a pharmacist at Bioluth. Bioluth is a company, a pharmaceutical company located in the south of France. Welcome. Very happy that you could be with us, Pierre, today.
Thank you to you for your invitation.
Thank you. And the third speaker is the Professor Jorge Saraiva, professor at Aveiro University in Portugal.
Thank you, Carol. It's a pleasure to be here.
So it's I will be introducing my co-presenter just before their talk. So now what I will do is I will start introducing the session with my first presentation, with my presentation. Okay, so I will be sharing my screen. Okay. I guess not starting at the beginning. Sorry about that. Okay. Okay, so I will give an overview of the HPP application in the pharmaceutical and cosmetics industry, what we can do with the technology. There are not a lot of application, even if the technology has shown an interest, and we can find the first publications on the potential application of the high pressure in pharmaceutical science, or I would say industry, and also in the cosmetic industry, that you can see they are from the turn of the millennium, so more than 20 years. The technology has an interest for cosmetic and pharmaceutical industry because some component of the raw material use or the final product of the cosmetic and pharmaceutical industries could be pressure sensitive or pressure resistant. Pressure sensitive, we have the vegetative microorganisms. The pressure acts on the membrane, on some key enzymes, can inhibit protein biosynthesis, which means that vegetative microorganisms can be inactivated by high pressure in cold condition, or at room temperature or at chill temperature. Concerning the membranes, biopolymer, and macromolecules, the pressure acts only on the three-dimensional structures, and some molecules are pressure sensitive, and the other ones, some others are not. So it depends really of the structure. In a general way, the pressure does not change the covalent bond. So from one macromolecules, we don't have several smaller molecule after HPP. We still keep the macromolecule. What we can change is the weak bonds or the non-covalent bonds. If the macromolecule or in general proteins, enzymes have a very complex structure, three-dimensional structure, or some aggregates, this can be changed by pressure. Has the small natural compounds that only have covalent bonds and not weak bonds are pressure resistant. The technology allows to keep flavor, aroma, vitamin, dyes, and some active molecules. So this means that the technology can be used to keep or to preserve these compounds. The nucleic acids are stabilized by hydrogen bonds, which are pressure resistant, so there is no modification on nucleic acid. And the polysaccharides, in general, they are pressure resistant. Some of them can be affected, so it really depends of their structure. So saying that, we can find 4 types of commercial application in the field of cosmetic and pharmaceutical products. Inactivation of biological agents, production of modified proteins, enzymatic hydrolysis, and cold and clean extraction of some biomolecules of interest. The first application, I will not go really in details because you will see that the next presenter will give much more input on those type of application. But the example you will see in the next presentation will show you how the technology can help to develop organic cosmetic products. In the case of Licolab, chemical free. So in this case, the HPP is the step is a processing step for the raw material. Could be for cosmetic, of course, could be the final product, but this is not the case in the case of Licolab. Bioluz, you will see that Pierre will give some details on the reasons why his company started with HPP. The technology in this case can increase the level of safety of the final sterile product. And the last, I would say, innovative or possible application will be developed by George from the University of Aveiro concerning the development of vaccine because the pressure can inactivate pathogens, keeping the immunogenicity. What we have to take in account concerning the inactivation of biological agents, of microorganisms with HPP is that the technology, when it is impact in the final packaging, need to be in a flexible packaging. We have launched 2 years ago a new technology which is the bulk. In the bulk technology, we are able to process before packaging the liquids. But if the products is processed in its final packaging, this final packaging need to be flexible, and the maximum volume for processing is a little bit less than 500 liter due to the size of the machine. The other things to take in account with HPP, limitation if you want, is that when the water activity is low, for example, for the dry formulas, the low water activity protects the microorganisms against high pressure processing, high pressure inactivation. So we are talking about liquids, formulas, liquids product, or semi-solid. We cannot process to kill microorganisms. We cannot process dry products. And concerning the last point, the bacterial spores, the pressure alone at cold temperature, chill temperature, room temperature, the bacterial spores are not inactivated by high pressure. In this case, we are working in a technology. We have performed at hyperbaric some tests with a very interesting, well, for me, I think, solution or technology proposed by Horst Ludwig when I was young, if I can say. I remember it was, so this has been published in 1997. And I remember when I read the first time this publication, for me it was like fascinating, because that was the first time I saw results. You can see on the drawing where at temperature between 50 and 70 degrees Celsius, that was possible. So they are really mild temperature, not cold, because at cold temperature it doesn't work, but with those mild temperature. In his laboratory, Professor Ludwig was able to show really several logs of inactivation of spores. In this case, it was Bacillus subtilis, used for pharmaceutical as a pharmaceutical indicator in the industry, which is now called Bacillus atrophaeus. And we have replicated those some of those trials last year, following the Ludwig process. And when we do pulses of 1 minute, 10 back-to-back pulses at 500 MPa, which is 5,000 bar, and going down, staying 1 minute and going down at 50 degrees, you can see the results we had in buffer. We did some tests on coconut water. When it was in oil, the results were a little bit lower. But as you can see with the orange bars, you can see that this could be perhaps a solution, not really in a cold condition, but with mild temperatures to inactivate spores. So another application, which is completely different, is the production of modified protein, protein with a medical or pharmaceutical interest. So this is in a range of pressure between 100 MPa, 200 MPa. And this is an idea that was patented. The patent was filled by a team of the University of Colorado, I think in Boulder, in the United States, if I remember well. Some people of this laboratory created or had a company called Barrowfold in Colorado, and they filled this patent application in 2006, and it became a patent in 2010. The idea is that you can see on the right on the drawing that when you process you produce a protein, if the protein aggregates with the high pressure, we can disaggregate the protein and obtain the native and the effective protein. So the technology allows to eliminate and refold protein aggregates. The result is that it reduces the immunogenicity of those proteins, and it increases the yield because you don't have aggregate protein, you have them separated. And the technology also enable to correct the refolding of the protein, because after synthesis, if they are not in the final conformation, the high pressure processing in this process that could take 1 or 2 days under pressure. This is very long. helps to recover the traditional or the natural configuration. And this allows to avoid chemicals, the use of chemicals in this type of processing step. After granting the patent, Barrowfold grants an exclusive license to Neuron Biotech. So it was 2010. In 2011, Naturon Biotech selected Merck Biomanufacturing, which is currently now Fujifilm, to start with one of our equipment, phase 3 clinical trials with interferon. So in order to refold the interferon. I have to say that I think this never goes more than the phase 3 clinical trials. I don't really know why. That's you. If you work with the pharmaceutical industry, you know that everything is confidential because it's so much investment in R&D, and so I don't know why this didn't went ahead at commercial scale, but it was feasible, sorry, and we had this application. That technology is interesting because Barrowfold sold all the patents, the IP they had, to a company in United States called Pressure PBI, or Pressure Bioscience Inc. And this company proposed to help the pharmaceutical company to develop this technology, so to go from aggregate to the native protein. And this is something you do not hesitate to contact them if you want to, if you are interested by this technology. PBI would do at lab scale, and if you want, we can provide the industrial machine to reach the commercial step. The third application we have of high pressure is enzymatic hydrolysis. The only example commercial example I heard about is about a technology that in Japan is called medium high hydrostatic pressure, so MHHP. So it's a pressure between 80 MPa, so 800 bars, and 2000 bars. And the technology helps to extract the enzyme, and also under pressure to increase the hydrolysis. And so this is an enzymatic hydrolysis assisted with pressure. So on the market, there is, or was, because I'm sorry about that, but my information is not really perhaps really updated, but there are some products, high quality placenta extract and collagen supplements that was commercialized after enzymatic hydrolysis under pressure. And the last example I have of commercial application is the cold and clean extraction of molecules. We have a customer that use the technology that use HPP to process vegetal raw material to be able then to use it and to enter as an ingredient in his process for a product that is delivered as an ingredient for the pharmaceutical industry. But unfortunately, this is confidential. So the only one that I know and is available on you can find information on on the website is the extraction of a bioactive component from muscle. In this case, what the high pressure does is that it opens the shell of the green mussel in New Zealand. So the process is the one required to be able to extract a marine lipid extract from the mussel, and this is a bioactive component, anti-inflammatory component that is used then by the company Liprinol for active component in their product. So as you can see, this is we have quite a lot of or very different application possible. So in conclusion, high pressure helps to develop products without heat or chemicals. It preserves small molecules. It's for the pharmaceutical industry. It can consider as a processing that has a quite low cost. It's very easy to implement. We have more than 300 machines all over the world implemented for food. So in pharmaceutical industry, we have no problem to install machine. It's environmentally friendly. Something I didn't talk about before, it's also we are able to keep with the high pressure the structure of the emulsion. And as you could see, a wide range of product could benefit from the technology. In any case, if you have any question, I would be very pleased to reply, or my colleagues from the application departments could help me also to reply to all your questions. Thank you very much for your attention. And now I will introduce the next speaker.
Hello.
Hello, Iker.
Hello, Carol.
Thank
Very
you
nice
to be with us.
To be here with you.
So I'm very pleased to that we have here Iker Portugal. Iker is a shareholder of the company Licolab in Spain. He joined Licolab in last year in the 2020 with the idea of transforming a scientific project into a business proposition. Iker has worked over 2 decades in different financial top management positions, including 11 years at the travel technology provider Amadeus. He's currently the CFO at 3P Bio, a CDMO biotech company owned by the French private equity group Kinside Capital. So Iker will today explain us why he used, or why his company used the high pressure processing to develop a very nice product.
Excellent.
The floor is yours, Iker.
Okay, thank you very much, Carol. So as Carol said, my profile is financial, so apologies because I don't know in great detail the technical aspects of the company, but I'll do my best to explain. So hello everyone. My name is Iker Portugal. I'm here to present Licolab. I'm the financial partner as well as a dear friend of Beatriz Uralde, who is the founder and soul of this wonderful project. Beatriz is a very clever biologist and a lover of the nature and sustainable projects. She has put a lot of resources and passion into Licolab. As you can see in the title of this cover slide, Licolab produces something very unique in the world, sustainable and natural collagen. I'm gonna develop this idea in the following slides. So next, if you go to next, if you go to the next, our Licolab, previous one, sorry. So a few words about Licolab. We emphasize, I should say that we emphasize the word natural because as you may or may not know, the collagen has always an animal origin. Collagen is used, as you know, in medicine and very often in very premium cosmetics in order to improve the effectiveness of the regeneration of the skin. The inconvenient is that the animals from which we obtain the collagen are always dead animals. There's no other way. But thanks to Licolab and Beatriz and the research, this is not always the case. Beatriz has managed to obtain collagen of excellent characteristics from a variety of snails in a process in which, and this is critical, the animal does not die or suffer in the process. This is completely unprecedented and very valuable for companies who want to use collagen in a natural and environmentally friendly products. We are, as far as I know, the only company in the world which is capable of producing this type of collagen without the animal dying or suffering. Licolab was created as a project of research and development, as many others, being a spin-off of research work at the University of the Basque Country. We are based in Vitoria, in the Basque Country, and we have developed into what we think is a great product for the use in cosmetics or the medicine field. Beatriz' innovative approach is part of the DNA of Licolab. So a few very simple milestones of the company is because the company has a very short existence, just a few years. All this started back in 2015 as a good idea, which won the Inicia Innovation Prize, which is a prestigious one. So a few years later, with some help, Beatriz creates Licolab as a legal company, very small, a tiny startup based in Vitoria, working at the technology park of Araba, which is part of the network of technology parks of the Basque Country. In 2019, she's able to produce as a product, as a final product, and ready for the market, this collagen. And 1 year later, this collagen is certified by the French certifier EcoCert as that we are a producer that in which the animal does not suffer, everything is natural, and we are 100% cruelty-free. It's not in this chart, but in 2021, Licolab has received officially further recognition for its sustainable activity, and I'm referring here to the carbon neutrality of its activity. So a unique product, as I mentioned before, our collagen is unique in the market. We obtained the collagen from a very specific snail type and through a very specific technique which allows to obtain very highly concentrated collagen with over 10% of concentration from the snail slime. And this is quite unique because for the ones familiar with the cosmetics industry, there are some others known and popular cosmetic brands that use snail slime as a core ingredient, but none of them has been able to obtain collagen from the snail. Consequently, the degree of effectiveness of those cosmetics to regenerate the skin is limited, unlike ours. So the specific technique that we use in the process to obtain the collagen is, of course, an industrial secret deposited in a public notary. In summary, our process requires to force the snail in a very gentle manner to produce a specific secretion, and then after that we can return the snail happy and alive to the farm, and in a few weeks the animal recovers, and it's possible to repeat this process all over again. During all this process, the non-cruelty with the animal is certified by EcoCert, which is very important for some companies. We are also very respectful with the environment. We do not use any very toxic or polluting ingredients in the process. We use HPP, high pressure processing, to achieve microbiological standards. We do not use any gamma radiation, as we think this methodology is not respectful with the environment, and thus is not compatible with our collagen, is not considered good for our products. If the next slide, please. In our market, I mean, obviously we are in the business-to-business market. We sell collagen to other businesses that use the collagen predominantly for cosmetics, and these businesses are willing to use our product because it's attractive both in terms of the intrinsic qualities. The collagen is very pure, very concentrated, has good properties, and at the same time, because it can be used with a natural and friendly, animal-friendly tag, allowing the manufacturer to sell, say, respectful cosmetics with this natural stamp by a prestigious recognized certifier as it is EcoCert. Our initial sales have been in the cosmetic industry, but we are trying to make a breakthrough. We have been accepted as a supplier with the purpose of incorporating our natural collagen to a medical process to heal and regenerate skin wounds. And so we are very excited about making progress in this area. So this chart represents, in very simple terms and quite graphically, our process. So everything starts from the careful process of obtaining, on the left, a particular secretion from the snail's slime. So the animal produces all sorts of slime, but not everything is useful, only just a particular one. So the resulting product is, which is the liquid, is treated with HPP, and later on, so we use the lyophilization as a key part of the process, as it allows the product to be stable and able to preserve its properties intact without any stabilizers, preservatives of any kind, or we don't need to, or they need to keep it cool. Our end product is a very efficient and very stable, so it's not sensitive to light or anything. So our product, in essence, is not very much affected by the HPP. However, allow us to have a final product without any risk, and obtain a number of certifications as we use HPP. I mean, the collagen is, as you may know, a protein that must be stabilized once obtained, and the sterilization must be used in order to be used both in the cosmetics and in the health sector. So the HPP allow us to reach the microbiological parameters required by the health authorities without damaging the functionality of the collagen molecule. The high pressures sterilize the sample without damaging the collagen protein. The pasteurization or any other method using heat will break the molecule, affecting negatively the functionality, and that's why we use HPP. Any other chemical component will take us, I mean, out of the natural an ecological equation. So that's why we use HPP. So next slide, please. So the formats, I mean, we in the cosmetic sector, we sell our collagen as powder. I mean, this is what we do is powder with very highly concentrated collagen in different formats, depending on what the end customer wants. And we are only with lyophilization is the only thing we do. We lyophilize the slime after the HPP process. Currently, we are doing some progress into the health sector, as I said before, to nanoformulate collagen to introduce it into a natural and ecological gel, which, as I said, tries to help heal some skin wounds for people with limited mobility who develop postural ulcers. In essence, the skin wounds. So we collaborate with companies from the health sector, as Biosasum, and some other groups of medical research at the University of the Basque Country. And we, in essence, we adapt the product to the needs depending on the applications which is directed to. So next slide. So why HPP? I think is straightforward. I mean, the quality standards and the ecological certifications that we need require that we look for innovative technologies that comply with both the effectiveness of the product and the natural values and sustainable development. Here, hyperbaric HPP help us to meet the environmental and the quality requirements and recommendations for the certification of our products, and also complying with the microbiological parameters required by the health organizations without altering the characteristics and effectiveness of our collagen. So that's why we use HPP. So the next slide. So we are in Alaba, in Vitoria, in the technology park, and due to the proximity in the area, we take ourselves the samples to the plant located in El Goibar, Aqua HPP Solutions, which is very nearby. So we can personally take the samples and bring them back in less than 3 hours, and thus we can, in the process, personally supervise that the cold chain and treatment of the samples follow the parameters established according to the good manufacturing practices. So that's why we like the proximity of Aqua. So basically, this is it. So thank you very much for your attention, and any questions you may have, I'm ready to take it.
Thank you very much, Iker, for this nice presentation. That was really great to hear about your company and your really nice product. I think this is a great application of HPP. We have one question for you, as you are in charge of the financial part of the company. Could you tell us what is the growth forecast you have for Licolab business? What do you forecast in the coming years?
Yes, I mean this is always a difficult question. I mean the financiers, we don't have a crystal ball, but we are optimistic. You know, we have started from a very small scale, and consequently, just growing from so a small base is always easier. We are learning in the process. We are taking some customers that like our product and use it in the cosmetics. This is a little bit, how can I say, little by little. So we would like to change the approach in the process. You know, it's not that we don't like that some people buy from us, but I think we are more in the line we acknowledge what we are. We are a research small company, so our talent is in the research and the scientific skill rather than in the manufacturing. We obviously manufacture, we have some equipment and so forth, but this is not our core strength. So we are looking for growth in someone that recognizes the quality of our innovation and is willing to partner with us. And the moment we identify this partner, I think Licolab is going to grow substantially. But we are patiently looking for the right partner. As anyone looking for a partner in real life, we are patiently looking for the right partner that understands our product and is willing to take it to the next level.
Okay, thank you very much. So
thank you, Carol.
And that's I hope you will find the right partner, and the company will grow very fast in the coming years. In any case, thank you very much, Iker, for your presentation.
Thank you, Carol. Thanks to you.
Thank you. So now I'm very pleased to introduce Pierre Combro. Pierre is the third speaker of this session, and hello, Pierre. I don't know if you could you hear me? Yeah. Hello. Thank you very much to be with us. So I would like to introduce Pierre, saying that Pierre is a pharmacist with a great experience in leading professional teams in the pharmaceutical industry. He is currently the CEO at Bioluf, and he joined the company in 2001, so 20 years ago. Last year, in 2020, Bioluf became the first co-manufacturer of pharmaceutical products for third party to implement HPP technology. So this is your company is unique in the world. Thanks to this strategic decision, the company Bioluf is now able to offer a unique value-added solution to your customer in the pharmaceutical industry, and that's what you will explain us in your presentation, and I'm sure this will be very interesting for our audience. Thank you very much to be with us, Pierre. The floor is yours.
Yeah, thank you to you, Carol, to give me the opportunity to present our company and to explain why we are for the moment alone. So yes, Bioluf is a CDMO company. We have roughly 11 million of turnover and 80 people, between 11 and 12 million euro of turnover, depending on the year. And half of the turnover is our own product, and the rest is the CDMO activity. We are only producing sterile product in flexible bags, and so it's a niche activity in the pharmaceutical industry. We are producing for human drugs, veterinary drugs, or medical device. And so we have lots of rule to follow for human, veterinary, and medical device, and it's it won't be easy to adopt HPP process for all part of the process in this activity. In this activity, we are specialized in the medium seria. Medium seria and small seria. There is a big player. We are doing million of flexible bags. We are producing roughly 5 million bags per year. And for small and medium seria, there is not a lot of player. We have decided a few years ago to invest in a new line to produce very small seria. We have some demand from our customer to produce some API for vaccine production or viral vector or for cell culture. and this production need to be done often in aseptic way. There is two process to have a sterile product. You can sterile when the product is finished, so what we call a terminal sterilization. And another process is to take a lot of precaution all during the process, and to produce and to work with only sterile products you adding, and so you are closing, and so if we consider if we are starting from sterile API and primary packaging, so we have at the end a sterile product. So you have some topic of validation to prove that's the case, but this is the general idea. There is different product concerned by this small area. It's medium for culture of micro or cell growing. Medium for media field test. It's the media field test is the test we are doing to prove that the process is doing in aseptic way, and there is a lot of buffer solution for biotechnology. This is the target of the line. And why high pressure? What I explained, to have a final product sterile, you have to take lots of precaution to have lowest as possible contamination, contamination before final sterilization. And so you are taking precaution for wearing, air treatment, validation, training, and filtration, and so on. And all this precaution, with all this precaution, we can reach sterility of assurance level. Next one. To start with a sterile product, you have different technology. E-beam, so gamma or beta, it's one of the technique used to start with, for example, API sterile. UV, it's very used for the decontamination of the surfaces of all material you are introducing into your isolator. The filtration, a very important point for the solution before the introduction into the primary packaging. Different chemical agents, and for the terminal sterilization, you are using sterilization by temperature, wet temperature or dry temperature. More often wet than dry. The wet temperature, the temperature of the sterilization in 120, and for dry it's more often 180. And high pressure is one of the tool existing to decrease the initial contamination of all our product using for the sterilization. And the end, when you are taking all this precaution and you are applying some of this technical or all of them, you have to prove that you have a sterile products, but so there is no reason that you don't have it. So next one. The aim, yes, the high pressure process, the application is interesting to decrease the bioburden, the initial contamination of the raw material. This is the first application we have, or to apply it in the finished product. We are working in flexible bags. So as Carol mentioned, high pressure process is indicate only in case of flexible packaging. Otherwise it's breaking. And so we can apply it in our flexible bags and for the raw material. And after the terminal filtration, we can apply the high pressure treatment on the finished product to be sure there is no contamination into the product. Next one. So the challenge, as I mentioned, is the validation of this process. So this technique is not used in our industry. It's very long to have the accreditation and authorization, but it's not because it's long that you don't have to try to do it. Because it's relevant. It could be one of the option we can use. We have not a lot of option to do aseptic filling. This is one of the possibility we have, and so we have to try it, and so to be sure to have the safety of the product at the end. So that's the end of the presentation, and so feel free if you have any question to contact us. You have the email, the telephone, and thank you very much for your attention.
Thank you very much, Pierre. That was really interesting. Your testimony is unique because you are the first. Not easy, but that's for sure useful. We have two questions for you. Is it possible to process all your products with HPP, or you see yet some limitations?
In fact, there is a possible yes, interesting not. If you are using terminal sterilization, it's not relevant to use HPP, of course. The only application in the case of aseptic filling, and all product. Yes, I think all product we can we fill in aseptic way could be we can apply high pressure process because it's a solution. It's an aqueous solution, so there is no problem for this.
Great, thank you. And another question for you is: do you expect to reach zero bioburden in 100% of your products? With in general, and I would say with the help of high pressure of HPP?
It's difficult to answer to this question because it depends of the initial contamination of the material. So what we expect is to have a decrease of 1,000 and 1 million. 10,000 and 1 million decrease of micro. It's what we expect. It will depend of the micro, as you explained. It won't be the same situation for bacil or for gram or moist. But so if we can decrease 10,000 to 1 million, it could be an interesting tool for the aseptic preparation. There is another very interesting topic for the application of HPP is the Nicoplasma. The Nicoplasma, so we don't like this Nicoplast in the biotech industry and in cell grow. So the problem of this of this micro is the size is less than 0.22 micro, and so all the filtration, the sterilization, the sterilizant filtration is 0.22 micro. So microplast, the filtration is not efficient for Mycoplasma, and there is no option except to avoid to have it at the beginning, but not sure to guarantee this before the end of the preparation. And so this application could be a solution to avoid this problem.
So you did perform tests on Mycoplasma with HPP, or and it's
In fact, yes, the Nicoplasma is tested in the product, and so what we propose is to do the test before and after. So the test of Mycoplasma, it's not easy. It's a PCR. It's difficult. But so there is some publication, and the efficiency of the HPP is good in Nicoplasma. So you can decrease 10,000 to yes, more than 10,000 decrease of the initial contamination. So that's the only option we have if there is a contamination for the moment.
So that's great that that HPP can give a solution for where there is no solution. So that's I
except
think
to
that's
remove the products and to restart.
Yes, but not really efficient. So that's a good example. Well, in any case, I would like to thank you very much for your presentation. Very interesting and very encouraging for the future development in the future use in the pharmaceutical industry. Thank you very much, Pierre.
Thank you. Bye.
Thank you. So now I'm pleased to welcome Professor George Saraiva from University of Aveiro. Hello, George. Could you hear me?
Yeah? Hello, Carol. Yes, I hear you.
Hello. So your presentation is the last one, and I would say the most innovative, because you will be talking about possible application in the pharmaceutical, cosmetic and pharmaceutical industry. So first, I would like to introduce you. So George has a PhD in biotechnology from the Portuguese Catholic University in Porto, Portugal, and is associate professor at Aveiro University in Portugal. His research is mainly dedicated to the use of high pressure for food preservation and processing, and for biotechnological application. And George will tell us about vaccines and also the possible application of a technology that I think you invent, which is the hyperbaric storage, which in some cases is not only storage, but could be also a technology to inactivate microorganisms. Well, I don't want to say more things. You will explain us. The floor is yours, George.
Okay, thank you very much, Carol, for the invitation and for the nice presentation. So it's a pleasure to be here. Yeah. So I will try to share my screen. So please see if you can see it. Can you see it?
We see it. Perfect.
Okay, great. Okay, so it's a pleasure to be here to speak a little bit of attempts of innovation we are doing with high pressure. So that's, I think, the most important job of the universities, to try to get new ways for new things, so for innovation. At the beginning, without thinking very much of the practical application, but to see if it really works. So I will speak about the possible use of high pressure for the production of vaccines. So that's and then and also after about hyperbaric storage. So in fact, if we see back, so high pressure is now used in the food sector for microorganisms inactivation. And we can see that high pressure can really inactivate the virus, some prions, bacteria, and fungi. So in speaking about this, about inactivation, we can translate a little bit or move a little bit for the pharmaceutical area, where some types of vaccines are vaccines where the pathogen agent is inactivated or attenuated. So it's able to generate an immune response, but still cannot infect or cannot infect a person and cause disease. So several tools can be used for these pathogens inactivation or attenuation, like temperature, chemicals, and so on, but also pressure can be used for this, with some advantages or at least some particularities related to the mode of action of pressure. So we can use pressure as a non-thermal approach, so to production of inactivated or attenuated vaccines, free of chemicals or at least with a lower level of chemicals, and that are able to produce a strong humoral or cellular immune responses. And here is very important not only the thermal aspect, but also the bulky aspect. Let's say, contrarily to or differently from temperature, where we have heat transfer problems and we have temperature gradients. Here with high pressure, we don't have pressure gradients, so we can treat bulky samples very easy, what is not very easy when using temperature. This is a very important particular feature of high pressure. So, for example, pressurized virus, we are suffering a pandemic problem worldwide. So, viruses are in fact being spoken everywhere. So, pressurized viruses are able to keep their immunogenic potential, mostly because high pressure can have some changes in their structure and not so much in their molecules. So, this means we can change their structures, like membrane, like the capsid, and inactivating them or attenuating them while causing much less changes on the molecules or the macromolecules or the macromolecules that are in this virus. And this keeps the molecules, the virus molecules, in a more, let's say, initial state and able to produce broader and with the potential to produce a broader and a more intense answers, so more intense immunization. So HPP can also be used, of course, in this sense, and Carol spoke at the beginning. Some of these changes caused by a pressure in virus and bacteria in the sense of producing vaccines are caused also in some proteins. So we can change recombinant proteins, as Carol showed, and also here we can also change some proteins of the virus or the bacteria or the pathogen agent we are speaking about. So, for example, we have some data already in the literature where HPP can fully inactivate influenza virus, to produce whole inactivated vaccines. This means very important. This is the whole pathogen microorganism that is inactivated and is used to produce a vaccine. In some cases, some vaccines are only produced with part of the pathogen organism, like some proteins or so. So the immune response will be really much not so much broader, not so much intense. And this is due basically to the formation of pores on the membranes and capsid damage. And like I said, in structures, in the architecture or the organization of the structures, not so much in the molecules. So very interesting to produce stronger immunogenic responses compared with split or subunit vaccines, and also the possible use reduction of some chemicals. And HPP is also very interesting because it can produce vaccines that keep the fusogenic activity. So because the virus proteinaceous structure is less affected, thus allowing the release of antigen in the cytoplasma, where the cytoplasma of the cell, where they are really processed and cross-present to T cells. So very, very interesting. And also with HPP has been shown that is two major immunogenic epitopes, hemagglutinin and neuraminidase, are better kept in many cases with HPP. And also sometimes a little bit related with what Carol spoke in the beginning about changing protein structure. Sometimes HPP can also induce the exposure of inaccessible epitopes, so that the immune response is really more stronger. So you have here some interesting data about some virus before and after HPP, and you can see some damages here. Very, very interesting for these applications. And for example, there are other possibilities in this area, like for instance, some results of on poliovirus, which are known to be resistant to HPP due to their small size, shape, and so on. And so, and also many times some live vaccines are usually very thermolabile, means these attenuated vaccines are very thermolabile. And we had this problem with the COVID vaccine at the beginning. Now it seems like this Pfizer vaccine can be stored much better at refrigeration temperatures, but anyway, they are very thermolabile. So what is the Sabino poliovirus vaccine? This is very important to this very the difficulty in the transport and handling in developing countries. So HPP was used to prove that using a previous high pressure treatment at 310 MPa for 65 hours, we could keep much better than the thermal conservation, thermal preservation of these vaccines. So this is the untreated vaccine. This is the treated vaccine as a function of the temperature, and you can see that this previous high pressure treatment could really increase the thermostability of this vaccine, which is very interesting for these cases. So HPP can enhance the thermostability of poliovirus without chemical stabilizers, which very interesting for several. It was studied at least for 3 types of poliovirus, and can also affect the thermodynamic stability of this poliovirus, and can be used to increase also the potential of and decrease the potential of vaccines. So very interesting. I think there's a lot of work, very interesting work to be here. We want to move on this field, and we are doing some efforts for this with a couple of partners. So hyperbaric storage, as Carol said, we are really very much involved on this since the beginning. So it all started as a methodologies or a pressure-based methodology that could compete or could substitute refrigeration, preservation of foods, but without refrigeration, so working at room temperature, so without spending energy to control the temperature, and so with no energetic costs. But then further, we moved this for room temperatures or above. So in fact, the hyperbaric storage concept can be used at sub-zero temperature, using the possibility of pressure to decrease the freezing point of water, and so have foods preserved at sub-zero temperatures without freezing and without formation of ice crystals. Can be used at refrigeration temperatures or at and above room temperatures. And we are mainly focused here because we avoid the problem and the constraints of having a refrigeration temperature. So we start to use this as an alternative to conventional refrigeration and possibly freezing, in a way that hyperbaric storage could serve as a hurdle to avoid or to slow down microbial growth as it occurs in refrigeration. But while doing these experiments, we start to see that maybe some other interesting features can appear, not only for reduced energetic costs, not also for reduced carbon footprint due to this, and so on. But we start to see that in addition to slowing down or being a hurdle to microbial growth as is refrigeration, we start to see in some of our experiments that some vegetative bacteria that we are studying were really inactivating during hyperbaric storage. And so we did a couple of papers on this. And then, of course, we start to think about what can be the case of spores, because as we saw in the previous presentation, for pharma and for food, spores are very resistant to several inactivation factors like temperature and so on, and then to pressure. So we start to study some bacterial spores, Bacillus subtilis spores, and we could see that in the low, poor nutrient media like macuvine buffer, we could see that if we study the total microbial load, like spores plus germinated spores, or only the spores, we can see that atmospheric pressure and room temperature, nothing really happens after 10 days. So atmospheric pressure refrigeration is also the same, nothing really very interesting happens. While if we increase at room temperature, we increase the pressure to 50 and 100 MPa, we start to see that spores don't germinate, but they even are inactivated. And we can see here nice inactivation levels, about 3 logs of inactivation. So very, very interesting, but this is a very poor nutrient media. Then we start moving further with the same spore, but with a very nutritive media, or a good nutritive media like carrot juice, and we saw a little bit the same, but with a small difference. That when we have atmospheric pressure, room temperature, in fact, the spores start to germinate, which is normal. And then under pressure, if we use 25 MPa at room temperature, we see also some increment of spores and germinated spores, while at 50 and 100 MPa, we don't see germination. We can see even inactivation of the spores. So you can see here total endospore loads inactivation of about 5 log, which is very, very interesting. Of course, this is a long time, but it's very, very interesting because it's a non-thermal procedure with potential to inactivate spores. So we move also to other types of spores, Alicyclobacillus acidoterrestris. So it's a spore that can resist pasteurization, for example, in acidic food products like apple juice. And they are pasteurized, the spore stays there and can germinate, and then can produce, in fact, likes very much to germinate in acidic media, and can produce guaiacol. So it's not pathogenic, but produces guaiacol, which is very terrible for the quality of the juice, and is also very resistant to temperature, very resistant to pressure. And we could see that in apple juice, when we inoculate these spores, we can see that at 150 MPa, we could see very, very interesting, even the 25 MPa, very, very interesting inactivation rates of and levels of this spore. While at atmospheric pressure and refrigeration, we can see basically nothing happens during this timeline. So we are also here seeing about something like 5 log reduction at a very important spore. So very, very interesting also for this type of spore. And recent work from some Japanese group of researchers, they used a very long-term hydrostatic pressure, so low pressure but very long, 2 years, so at 4 degrees Celsius on different viral particles, and they are a little bit confirm what we know about the possible use of pressure for inactivation of virus. This is a kind of a very long hyperbaric storage, and they conclude that when the virus is very small, or for smaller virus, the structure and this can be kept intact even for this time, and then they are able to grow and to multiply. While as we increase the size of the virus, we can see progressive inactivation of this virus. So they also found some interesting differences for the different types of virus and different types of capsids and so on. This is very interesting for the future to be studied. So very, very interesting to further study this. So I think it's about to reaching my time. So this is the group of researchers who basically are working more directly with me. We have also some other collaborations with students that are not here. And so if you have any questions, I'll be very delighted to answer to them now or by email. And in this QR code, you can get some more information about high pressure research for these areas and about our Innovate group at University of Aveiro. Thank you very much.
Thank you. Thank you so much, George. Very, very interesting presentation. Amazing possibilities for vaccine and also for spore inactivation at with the hyperbaric storage. George, I have a question for you. I would say I have the COVID question. Do you think HPP or hyperbaric storage could be used to develop COVID-19 vaccine?
I think it is interesting. It is interesting to try because it's a different virus, so not really much. Although the effort being done on research to produce vaccines is very recent, so there is not much information. So we don't know what happens with pressure. So we are thinking about on this to start some research on this with some partners here. But this is very interesting. It will be very interesting to do some trials for sure.
Mm-hmm. So that would be great if in the next presentation or next conference you would be able to show that, because COVID-19 is the hot topic. So
Yeah.
You will become very famous
Yep.
if you can publish on this virus.
Let's see.
And what do you think could be the first application for hyperbaric storage? I mean, could be food, pharmaceutical, cosmetic. But what do you think? The first that could show up or could make the technology commercial, a commercial stage.
I think at this point could be two types of things, independent of being pharma or food. The first is to have low volume products with high value, so that we can do hyperbaric storage for this on a smaller pressure equipment. Yeah. And the second one is to have some products that by HPP they suffer changes in quality, while by hyperbaric storage you might reach similar inactivation levels without changing some interesting, let's say, sensorial properties.
Mm-hmm.
That's mainly the two lines we are studying here with a couple of interesting cases. We have 2 or 3 papers published on meat.
Mm-hmm.
Yeah, and if you see, we can preserve meat, have some microbial inactivation by hyperbaric storage without really much effects on color, which can be very interesting for the food area.
Mm-hmm.
But we need manufacturer suppliers like hyperbaric to start thinking about to develop equipments for high volume hyperbaric storage experiments.
Mm-hmm. So
that would be very interesting.
Now the challenge is for us. So
yeah,
if we cannot depressurize the machine for, I don't know, 2 weeks, 3 weeks, or 3 months, that's a challenge for the industrial equipment.
I know.
That's great. So we have work for the future.
Yeah, sure. I think you have work a lot, and a possible new field, I think.
Hope so. So thank you very much, George, for your contribution to this session. Very interesting.
Okay.
And it's now time to finish the session. So thank you for your attention, and we were very pleased to that you listen this session, which is a little bit outside of the food topic we have in the rest of the session, but I think open new application, and could be also interesting for the food business, especially the presentation on the hyperbaric storage we just saw. Thank you very much.